Great. Can everyone see that?
Yes.
Okay, good. You know, I always like to keep my talks as informal as possible. So if you want to unmute yourself and interrupt me with a question, please do so. Don't have to wait till the end. So I want to thank everybody for their invitation. It's nice to be able to talk about something other than SARS-CoV-2 even if it's for an hour. We have been in the background kind of trudging along on our HIV work, and we're excited to share with you kind of the latest that's coming out of our lab. So as a lot of you know, our lab is a systems biology lab. We look at host pathogen interactions using various omics technologies. And I'm going to tell you about two stories that initially started as RNAi screens, but we've really driven down into understanding specific factors that came out of those screens and their impact on HIV biology. So the first story I'm going to tell you about is something that Sunny Yo has been working on probably for the better part of the last 7 to 8 years, along with a number of collaborators. So she published her initial findings on this project back in 2015, and it really stemmed from an observation that Dan Littman published and others have made, including our lab, that if you challenge HIV along with HIV-2 or SIV-VPX, if you challenge myeloid cells with those two constructs, or the virus and a VPX, VLP or a protein, you could elicit an innate response. So in this case, we're looking, I'm not sure if you can see my pointer here, but we could see an innate immune response. This is ISG54 mRNA, which is an IRF3 target gene. But if you look at nearly any IRF3 target gene, you can see that there was an innate response to HIV. If you add nevirapine, which is a reverse transcriptase inhibitor, as you know, you can block that response. And if you add an integrase inhibitor, raltegravir, you dampen that response, but it's still there for the most part. And this was really the first time that we saw HIV really triggering an innate response in any cell culture model. And so we were pretty excited about this. This was in primary human monocyte-derived dendritic cells. And what Sunny did was go ahead and set up an RNAi screen. So this was not a genome-wide RNAi screen, but we focused on potential nucleic acid binding proteins. So there's about 500-600 of them. And what we did was knock them out and essentially asked which one of these genes could extinguish this immune response we were seeing to the virus. And we found a handful of them could, and she quickly drilled down on this one protein called PQBP1. And just to put this into context, when we were doing this was before the elucidation of CGAS as a sensor of HIV DNA and other cytoplasmic DNA. But I'll talk a little bit about how this project dovetails into that. CGAS. I won't go into the details, but I wanted to give you kind of enough of a background on this so you'd have better context into the new data. Again, this was all published back in 2015. So this PQBP1 protein is actually a protein that's been implicated in neurological development. It's mutated, and I'm showing you some of the mutations. There's an insertion, deletion, and point mutation in what's called Ren-Pinning syndrome. So this is a developmental disorder. We had absolutely no idea, still don't have any idea what that patient population may have to do with immune sensing. But through a lot of work, one of the most interesting observations is if you take myeloid cells from these patients and challenge with HIV, what we're finding is that they don't respond to HIV in any sort of innate immune capacity. Whereas, of course, as I showed you before, normal patients do. And so The finding of that paper was that PQBP1 was an essential cofactor for CGAS to recognize reverse transcribed HIV DNA. That allows CGAS to synthesize cyclic GMP, which is a mediator of STING that induces interferon-stimulated genes in type 1 interferon. If you knock out PQBP1 and look at CGAS activity, CGAS makes this second messenger cyclic GMP. PQBP1 was absolutely essential for generation of CGAMP synthesized by CGAS. So the interesting thing, the other interesting thing about this molecule was that it seemed to be exclusively required for retroviral DNA. Right here, I'm showing you HIV-1, but we tried other retroviruses, and PQBP1 was required for the immune response to retroviruses, but not other DNA viruses. In this case, I'm showing you MHV68. But if you even transfect in DNA, we don't see it's required. We also, Sunny also showed that PQBP1 binds HIV DNA, but not mitochondrial DNA or DNA from other viruses. So after that paper, we had this finding that there was a retroviral specific cofactor for CGAS that's required to recognize reverse transcribed HIV DNA and initiate an immune response to HIV. But there were a number of open questions. The first one being, why does CGAS need a receptor, a co-receptor for HIV DNA and not for other kinds of DNA? And a second question, and I think probably two sides of the same coin, is really how does CGAS PQBP1 get activated by one or just a few copies of transient DNA products that's generated by HIV? So in contrast to other DNA viruses, which is what this cartoon shows, other DNA viruses make hundreds of, if not thousands of copies of DNA that go through the cytoplasm that enabled potential detection by a cytoplasmic DNA sensor. But as you all know, HIV only spends a very small portion of its time as a DNA virus. So essentially from reverse transcription until it integrates, at which point it's camouflaged from any sort of sensing mechanism. And typically you only have one viral infection per cell, maybe a couple. The question really we were trying to ask and answer was that how does a virus, how does the host immune system really capture that transient DNA species, and how does it distinguish that from any other DNA that may be floating around in the cytoplasm, from mitochondria, from DNA leakage, from the nucleus? This is a careful balance that the immune cells need to make, because you don't want to trigger an immune response to self-DNA, which of course leads to autoimmunity, but you still want to be able to exquisitely sense a small amount of DNA species coming from a foreign DNA virus. And so through collaborations we've done with Tom Hope and some other groups, we've began to start to unravel that question. So the first clue came when we were looking at PQBP1 association with incoming HIV. So we wanted to see at which point does PQBP1 associate with DNA, HIV DNA by immunofluorescence microscopy. And what Tom and Zhao Mehmed and Tom, who was at Tom's lab, he has his own lab now, was able to show is that PQBP1 actually interacts, well not interacts, but at least co-localizes with incoming virions, right? So quantitatively, about 50% of the virions are decorated by PQBP1 when you look at it through immunofluorescence. We then teamed up with Till Boking in Australia to see if perhaps does PQBP1 associate with the HIV capsid beyond just our association with DNA. So this is a kind of a quirky biochemical assay, but it seems fairly powerful in that what he does is he labels the capsid with a fluorophore, and then he labels your protein of interest with another fluorophore, and you essentially float that along a focal point. And if you see that if the two proteins interact, those two fluorescent signals will temporarily emit a coincident fluorescence. And so what this cartoon is showing you is if PQBP1 and capsid were actually interacting, you would see these kind of fluorescent bursts at this focal point in a coincident manner. And so he can show that with CYP-A co-localization to capsid. If you take assembled capsid, co-incubate it with CYP-A, you see this high level of co-localization using this assay that's called SMS. And if you add cyclosporin, which disrupts this interaction, you can extinguish that. And what we find is full-length PQBP1, whoops, sorry about that. Full-length PQBP1 gives us a similar co-localization pattern. We did some structure-function mapping, and we found that the N-terminus is what was mediating this interaction. If you chop off the N-terminus, that interaction goes away. And if you just express this N-terminus, you can see that this is what's mediating that level of interaction. And so we've also done some biochemical analysis on this, and we see similar kind of data with in vitro synthesized capsids. The next piece of data kind of for us that convinced us that this was really was going on was some FRET assay where we labeled CYP-A with DS red and PQBP1 with YFP, and we asked if there was FRET between PQBP1 and CYP-A on the capsid, and essentially that's what we're seeing. We're seeing a high level of FRET between PQBP1, and this is the N-terminal portion here. I'm showing you PQBP1 and CYP-A at the HIV capsid. Again, suggesting that PQBP1 is directly binding to the HIV capsid as the virus is coming in after entry. And this is the quantitation of that data there. Okay. So then functionally, we want to ask, is this interaction relevant for immune response? So what we did was we knocked down PQBP1 in these cells. So these cells, you can't CRISPR them out. This has kind of been a bane of our existence. It's lethal to these cells. So we have to do a transient knockdown and a reintroduction. But anyway, if you knock down PQBP1 and then you put back in the wild type, you can reconstitute immune induction. But if you lop off the N-terminus domain of PQBP1, this is the domain that mediates the interaction with capsid, we can no longer see that immune activation. So this was biochemically a domain that we saw that was necessary for capsid interaction, and also functionally something that was necessary for the immune response. So we concluded that the interaction between PQBP1 and capsid was required for immune response. The other interesting piece of data is that if we express just the N-terminal fragment, so none of the downstream end, but the fragment that of PQBP1 that interacts with capsid, it actually acts as a dominant negative. So we postulate that the N-terminus is just overexpression of the N-terminus is competing away wild-type PQBP1, which is, and the C-terminus is what interacts with CGAS and allows you to get the immune response. So if you just overexpress the N-terminus, you see the squelching of the innate response to HIV when you measure ISG54 or any other immune response. So this doesn't happen with Sendai virus or other innate immune stimuli. So, and again, the next thing that we wanted to ask was where does CGAS come into this? So we were fairly convinced that PQBP1 is recognizing HIV capsid as it comes in. It's recognizing it through the N-terminus. We also knew that PQBP1 was required for CGAS-mediated immune response. What's the link here between the capsid recognition and CGAS? So this is still not a complete story, but I think we're getting close. And I'm sharing with you some of this data. Some of it was nailed down, some of it preliminary. But what we find is that PQBP1 and CGAS have an increased co-localization after HIV infection. And this doesn't happen if you just transfect in DNA. So just inducing CGAS by itself doesn't induce a co-localization between PQBP1 and CGAS. It's actual infection with HIV that does this, and this co-localization happens at HIV, at the HIV capsid or HIV particles. And we can recapitulate this biochemically. So we see very weak interaction between PQBP1 and CGAS in the absence of HIV, and you can see that interaction significantly gets increased after HIV infection. And so temporally, how is this happening? So again, working with Tom Hope, we used a nifty little system that he's developed where he fuses or loads integrates with a red signal, and then he produces HIV in the presence of GFP, so that GFP gets incorporated both into the virion as well as the core. And when the virus fuses, you're able to track that because you lose one level of GFP signal, and then when you lose capsid integrity, you lose a second level of GFP signal. And so you can temporally trace when all of this is happening. And so what we did was look to see when PQBP1 and then CGAS gets recruited to the virus. And what we find is that on the left side, PQBP1 is recruited when the virus fuses, so after entry, and then stays on and after integrity loss. So this is the initiation of uncoding and reverse transcription, whereas CGAS really only comes to the virus after the initiation of capsid uncoding and reverse transcription. So we concluded here that PQBP1 goes to the capsid first when it's intact, and upon the initiation of reverse transcription, we have some other data that I'm not going to show you that suggests that it is either capsid integrity loss or the initiation of reverse transcription, or both. It's really hard to separate the two. That's triggering the recruitment of CGAS. So the next question that we're trying to answer, so sorry, this is just the amount of PQBP1 that's positive. You see a lot more PQBP1 that's positive than CGAS, but almost all of the CGAS that's recruited is recruited in conjunction with PQBP1. So the reason I have an asterisk on this this slide here is we're still not happy with this data, but we're trying to establish that PQBP1 is required, absolutely required for the recruitment of capsid. So I mentioned to you before that we can't make a knockout of PQBP1, a stable knockout with a CRISPR. The cells all die off. This is something that's absolutely necessary. And so what we've been trying to do is make transient knockouts, okay? But these are not clean. The cells stay alive for like 5 or 6 days, and then they just peter out. And so this is kind of what that transient knockout looks like. So the red here is the wild type level of PQBP1. The blue is how much knockdown we get during this transient knockout. You can see that there's still some PQBP1 left, so it's just not a very clean assay. And so this is why we're not able to get really clean data. So we're trying to clean this up, but the initial data, and this is again preliminary data, which is why it's asterisk, we see that knocking down PQBP1 hinders the recruitment of CGAS to the capsid, okay? This is not statistically significant, hence the asterisk. So we're trying to come up with other strategies to nail this down, but at least we've done this enough where the trend is going in that direction and is strongly suggestive that PQBP1 is actually mediating the recruitment of CGAS to the uncoding capsid. So this is our model that we have so far. So we call it the two-factor authentication model, right? So getting back to the original question, how does one copy of HIV DNA really trigger a response to the innate response to HIV? Well, our answer to that question here is that HIV is first checked, that DNA is first checked by PQBP1. PQBP1 is decorating the capsid, and that's the first authentication level, right? It's kind of like on your iPhone where they make you put you in your password and they send you a text and you have to authenticate it's you. PQBP1 is the password, and PQBP1 is required for the recruitment of CGAS and the recognition of DNA before you trigger that immune response. So this is how we believe that the cell can authenticate a single copy of DNA, concentrate CGAS onto the incoming capsid, and then trigger an immune response to a low-level PAMP that may be an inauthentic PAMP if it weren't for its authentication by PQBP1. So that's where we are in that story. I'm going to pause for a second, so I'm going to completely switch gears to HIV latency. But if anybody has any questions around that story, I'm happy to field those right now.
Hey, Sumit.
Yes. Hey, John.
Hey, this is John. I was just wondering, you know, always bothered me a little bit about the fact these are monocyte-derived dendritic cells, right? Is that right?
Yes, they are.
So for HIV, of course, VPX had to be used to reveal this. So those cells would affect. The point was to get rid of the restriction by SAM-HD1 in those cells. So what happens to authentic HIV when it doesn't have that? Is this would be acting as sensitive?
Yeah, this is a great question. So we've been trying to figure this out for a long time. So you can use VPX, or you can use an obnoxiously high concentration of virus to elicit this response, right? Nothing you would ever see physiological, right? So what we surmised is that this is a rare event, okay, that happens, and potentially a rare abortive event. And so we've been doing, Sunny's been doing some single cell analysis of this phenomenon, plus minus VPX. And this is what we see, right? It's not that all cells kind of respond evenly at a low rate. It's just that there are very few cells that respond at a very high rate. So, you know, 1 out of every 1,000 cells will have an immune response to HIV, even with VPX, right? VPX just kind of juices that up. I think what's happening is VPX is not only enhancing infection, but enhancing abortive infection, right? I don't have any,
right.
We have some single cell data that's very suggestive of this, but I don't have anything to nail that down. So that's our working hypothesis, is that this happens rarely, which is why HIV, you know, causes its disease, the disease that it does. Because if it was aggressively recognized by the innate immune system, you know, it would be stopped at the mucosal barrier, right? So, you know, it really flies under the radar, except in kind of very rare circumstances. So the immune system is not able to mount an aggressive immune response until you have high concentrations of virus. So I think this is why you see interferon in patients once viremia has been established. but not kind of during the initial stage of an infection. So that's a lot of hand waving. I hope that the single cell data, as we progress, will shed some more light on this hypothesis, either rule it in or rule it out. But yeah, this is kind of the system that we're using. And again, it's not VPX dependent. You can do it with high concentrations of virus. But again, that has its own caveats and flaws too.
Cool, thank you.
Yep. Okay, if there's no other questions on this piece, I'm going to switch over to a latency piece. I don't have to really educate this audience on latency and shock and kill, but really in a nutshell, you know, latency reversal, people think that basically waking up the virus with a shock agent and then either relying on the immune system to suppress viral replication or eliminate infected cells, or even adding a kill agent like a CAR-T can eradicate the HIV reservoir. Unfortunately, the known latency reversal agents to date have not been done well in clinical trials. PKC agonists, which activate NF-kappa B, leads to T-cell activation. HDACs, which can also reverse latency in cell culture, actually are not that potent and have suboptimal efficacies. And so the goal here is to get the kind of the Goldilocks latency reversal agent that doesn't induce immune activation, but it can induce latency reversal at sufficient enough levels. So this story actually started when I first set out my lab here in Burnham. Some of you may remember a genome-wide RNAi screen that we did looking at HIV host pathogen interactions. Lars in the lab decided he wanted to look at the flip side of that data set, and so not look for things that are required for HIV interaction, but things that actually increase HIV infection, right? And so the right side of this panel, you see these are all the genes that he was able to confirm by 2 or more siRNAs increase HIV infection in cells. We work with Viviana Simon to publish a story on CYLD. But today I'm going to talk to you about this protein we've been working on for a while now called CIAP. I'm going to use that interchangeably with BRCA2, but they're essentially the same protein. What Lars was able to show is that knockdown of HIV actually enhances HIV transcription and not reverse transcription or integration. We teamed up with Nick Hosford, who's an incredibly talented chemist here at Burnham, to come up with a drug that targets this protein CIAP1 for degradation. It also targets CIAP2, and you can see what this protein what this molecule does by Western blot. Here, our genetic data suggests that BRCA2 CIAP1 it's what's mediating most of this event, but just something to keep in mind that this molecule that we're using causes degradation of CIAP1 and 2. But we also see latency reversal that accompanies, this is in JLAT cells here, that accompanies degradation of this protein, essentially chemically validating what we saw with our siRNA. So what is CIAP1? So it's essentially a suppressor of the non-canonical NF-kappa B. I don't know if many of you may have worked with Alex Hoffman, who used to be at UCSD. A lot of you have known the canonical pathway, the non-canonical pathway. It's essentially the slower, more deliberative cousin of the canonical NF-kappa B pathway. It's not the sledgehammer that canonical NF-kappa B signaling is, and this is what piqued our interest. So what happens is that CIAP1 leads to the constitutive degradation of NIC, and when the pathway is triggered by a ligand, either CD40 or LT beta. You see degradation of CIAP1, stabilization of NIC, which is what you see in the Western blot here, and then processing of P100 to P52, which teams up with REL-B, goes in the cytoplasm, into the nucleus, and triggers an overlapping set of genes that overlap with NF-kappa B. But again, it's not the acute induction, it's more of a slower induction that's more long-lasting. And what the molecule that we have does is essentially binds to CIAP1 and triggers its autoubiquitination and degradation. So it's a family of proteins known as Smac mimetics. And so these Smac mimetics typically are used to enhance trail-induced apoptosis, but CIAP1 and CIAP2 are the only two Smac mimetics that actually are known or have been reported to function on this non-canonical NF-kappa B pathway. And most Smac mimetics have some level of activity against all of these, or at least a subset of these Smac-related proteins. So this has all been published in largest cell host and micro paper, but what he was able to show that this molecule, the Smac mimetic that we had, that was directly targeted at CIAP1, this is in patient cells from ART-suppressed patients, and study done in Vicente Flanellis' group. By itself, it had some meager reactivation activity for HIV, but if you combine it with panabinostat, which was an HDAC inhibitor, we saw reactivation activity comparable to CD3, CD28. Okay, so this suggested to us that one, this mechanism was worth looking at, and two, the molecule that we were working with probably suffers from a lack of potency, and if we could optimize a better molecule, we may be able to focus ourselves on a therapeutic. And we felt that at least the mechanism of action was validated. Now we just needed to move into kind of more of a drug discovery mode to optimize and test this. And so what you're seeing here is a bunch of SAR and medicinal chemistry that allowed us to optimize our original compound, which is in blue, to our second generation of compound, which we are calling Ciapavir, because it's a degrader of CIAP1, and it's much easier to say than SBI 0953294. So for here on out, I'm going to be calling this molecule Ciapavir. We were able to show that this molecule still works through the non-canonical pathway. So this is latency reversal in JLAP. And then if you knock out one of the members of the non-canonical pathway, which is NIC, you see that this latency reversal activity goes away. Again, this optimized molecule we think is working the same way, but just a lot more potent than our monomeric molecule. This molecule also was clean as far as immune induction goes. We saw, if you add it to resting CD4 positive cells and look at CD69 or CD25, we don't see anything. We also don't see any cytokines. I'm not going to show you that today, but that's data that we recently published, I think last month or the month before, in a Cell Reports Medicine paper. So the next step is to see if this worked in vivo. And so we had to look at PK/PD markers. And so we optimized this for half-life also. So this has a significantly greater Cmax than half-life. So our half-life was coming in at about 3 hours with a Cmax of about 8 micromoles per liter. And if we look at PD markers, so our PD marker here was CIAP1 degradation, we could see at 10 mg per kg, we can trigger degradation in the mouse in both the spleen and thymus of CIAP1. So this gave us reasonable hope that this would work in vivo. So the first thing we did was we put this into mouse at 10 mg per kg and 20 mg per kg. First thing we did was look at cytokine response. We wanted to make sure that this was clean in vivo as it was ex vivo, and we didn't see any cytokines coming up. This is just a snapshot of the cytokines that we looked at. And then we teamed up with Jerry Zach and Matt Marson. Jerry's at UCLA. Matt used to be in his lab. Now he runs his own lab at UC Irvine. Most of you are familiar with this BLT model, but essentially it's a human thymus liver CD34 stem cell transplant into mice, then challenged, infected with HIV, suppressed with ART for about 6 weeks, and then we treated with Ciapavir here to look at latency reversal. And so this is the data that we got. So at low dose Ciapavir, we see 1 out of 3 mice, which is shown in yellow here. And at higher doses, we see 3 out of 6. So if you look closely at the data, it looks like the mice with higher viral loads could be reactivated better than the ones with low to undetectable viral loads. And we included all of this data, but this is a tricky model to work with. But we think that these mice just couldn't reactivate because they just didn't have high enough established reservoir. And if we look at bone marrow, so what I showed you before was plasma levels. If you look at bone marrow at the high concentrations, we see 4 out of 6 mice. And these 2 mice that didn't reactivate, we had very low viral loads, so max viral loads detectable about plasma viral loads before ART suppression was less than 10,000 RNA copies per ml. Again, that's hand waving, but it seems to track with the ability of this molecule to reactivate. So this is just one of the shortcomings of the model, but you know, in many ways for us, we were excited because now we had some in vivo proof of concept that this mechanism that we showed in vivo, ex vivo could work. And while we were putting this together, this is just some in vivo CD69 data showing that we don't see a lot of immune activation. And there's also in the supplemental in the paper, we've done a panel of cytokines. So this is relatively inert. There's some markers that go up slightly, but nothing that we would consider that would be dose limiting tox. And so while we were doing all of this, we knew that the Margolis Consortium over at UNC and Victor Garcia has been working on another molecule. It's an AstraZeneca molecule that has a very similar mechanism of action as Ciapavir. And in, I think it was either late January or February, they published a study showing the same thing, showing that they were able to see reactivation in a mouse model, a BLT model, where they were able to show about 50% of the mice at various doses can reactivate HIV. And then they moved to an SIV model in monkeys. And this is something that you know, we at the time did not have access to, and showed that this molecule was able to reactivate HIV or SIV fairly consistently without any real high levels of immune response. And again, underscoring, I think, in vivo and very much in line with our data that suggests that at least this mechanism is continues to be an attractive approach for latency reversal. So there are a whole number of liabilities of this AZD molecule. It was shelved by AstraZeneca for a number of different reasons. And I've heard that the Margolis group is going into clinical trials, not with this molecule, but some other molecule that they haven't yet published on or haven't released. And so I'm not clear as to what they're doing clinical trials with. But the latest on our molecule, Ciapavir, is this. So we teamed up with Chris Peterson and Hans-Peter Kim up in Washington, and did a pilot study, not using SIV, but a SHIV model. The SIV model, I'm told, I'm not an expert in HP biology here, but the SIV model is much easier to reactivate than a SHIV model. But what we found is for us, in the SHIV model using Ciapavir, 3 out of 4 of these monkeys were able to reactivate virus well over the limit of detection, anywhere from 60 to 100 copies of viral load. You have to remember, these are still ART-suppressed animals, so I'm told that this is a reasonable level of induction. So the interesting thing is that the AZD molecule did not read out in this model, right? So it wasn't really a head-to-head comparison, but the AZD molecule did read out in the SIV model, but not in the SHIV model. We haven't ran our molecule in the SIV model, but again, I'm told that the SHIV model is a much higher bar to clear. So we think that the Ciapavir is going to be a more efficacious than the AZD model. And so we're excited to expand this study, and we're expanding this study with Chris Peterson and Hans-Peter Kim, and putting in a potential kill agent in the form of a BNAb to see if we can also get not only reactivation, but reservoir clearance. So that's the latest update. So this latest monkey data only came out a couple of weeks ago. That's unpublished. Again, pilot study, but pointing in the right direction. We hope that a larger study will bear out some of the data that both Victor Garcia saw and we're seeing in this initial pilot study of 4 animals. So just wanted to thank the folks who did the work. I mentioned Zhao Mehmed and Tom Hope's lab, Derek Lau and Till Bohing's lab, and Maria Sanchez and Adolfo Garcia Saucer's lab, who did work with Sunny on the innate immune story. And then, of course, Lars, Nick Cosford, who developed the Smac mimetic in collaboration with Peter Trietti. Jerry Zach did the BLT model, and Vicente Planellis did a lot of the ex vivo work. So with that, that's my last slide. I'm happy to take any questions on this piece of the work, or if something came up from the innate immune piece, also happy to discuss those findings. Thanks for the applause, John.
You're welcome. Great talk.
Thanks.
Can I ask a question?
Of course.
It's just so quiet. So this is actually a protein, this molecule. It has to be given like by injection, or
yeah, it's not orally bioavailable. So it's a cyclic peptide. So it's
right.
Still a small molecule, but it's not orally bioavailable. So the first generation molecule that we had was orally bioavailable, but we just couldn't optimize its potency as an oral formulation. So this is going to need to be, if it does go into the clinics, it's going to need to be an IV type of therapy, which I don't think for this. I mean, I'd be happy to kind of discuss this, but for this kind of application is not a deal killer, right? I see people going into the clinic, you know, maybe for a 1 to 2 to 3 month kind of treatment of this with a kill agent, and then, you know, establishing a cure. It's not going to be something that you're going to be doing chronically, that, you know, you really need to be taking a pill for. But, you know, I'd be happy to hear other arguments, especially for those who actually work with patients and have maybe stronger insight into that than we might. John, what do you think? Do you
Well,
think,
I wouldn't think it's a deal killer. I mean, you know, intermittent intravenous therapy, if it cures HIV in a few months, I mean, shoot, that seems fine.
right? That's what
That's a
that's
deal
what I'm
killer.
guessing, but yeah, that's where I'm guessing, but you know,
Did you,
I've never seen a patient, so I'd leave it to you guys who actually see patients to tell me I'm wrong.
did you had synergy with the HDAC in the ex vivo data? Is that something
Yes.
you're planning to try in the non-human primates as well?
Yes, we want to try that in the not. So we had synergy both with the BET inhibitors and with the HDACs, and so we're trying to push them to try those. But you know, again, these are unfunded studies, so we kind of have to pick and choose. And so right now we decided to go. Well, they decided to go. They kind of said, hey, we want to try your thing with a BNAB or nothing at all. And so we tried to ask them to say, hey, let's do some synergy studies in vivo. Hopefully we can get a grant to do some of that work, because I think that, you know, that'll really boost up some of the LRA strategy. But that's on the to-do list, both the BET-Is and the HDAC. And we're doing other screens to look for synergy. So I don't know, John, if you saw our SARS-CoV-2 study, the drug screening study that we put out with that reframe library, which is 12,000 known drugs. We're running that library. So Vicente is running that library to look for things that can reactivate HIV and then reactivate it in synergy with Ciapavir. So the nice thing about these is these are all going to be clinical stage compounds. So if there's anything else out there, right? We tend to be looking under the lamppost for things that are reactivating agents, right? And I think I like the strategy in that, you know, it's going to take almost all of the known drugs out there and see if there's anything there that we didn't anticipate that might have that, and then start going into these. So unfortunately, the BLT model, we can't write another grant for, well, depending on what happens in November. But for now, we can't write another grant for the BLT models. But the NHP models, hopefully we can get some funding to be able to test out some of these in these combinations.
Thank you.
Yep. Any other questions? All right, well, if you guys think of anything, you feel free to email me, but thanks, that was great, I enjoyed it. And I'm gonna, if there's nothing else, Kim, I'm gonna sign off.
Thanks, Sumit, that was great.
Oh, thanks, Davey. We'll speak soon, everybody. Take care.
Yeah.